Recording is on.
All right. Okay. All right. So once again, thank you for inviting me to give the second ESID Juniors Journal Club. I'll be presenting my paper entitled Erythropoiesis Defect Observed in STAT3 Gain-of-Function Patients with Severe Anemia. That was published last year in the Journal of Allergy and Clinical Immunology. And thank you again to Clara and to Sandra for organizing everything. In the spirit of ESID Juniors Journal Clubs, I hope that everyone feels open and interested, and that afterwards there'll be lots of questions. There's no questions that are too stupid, and I hope that I'll be able to answer the questions as well. It's been a while since I've been in research. I've been in the clinics now for 2 years, but I hope that I'll be able to answer everything, and if I can't, then I'll be sure to get back to you with the answer in due time. So I will try to tell you how this paper a little bit came to be as it actually realistically happened within the lab. Sometimes when you read a paper, it all sounds like everything perfectly fell into place. And I think what's interesting, if someone presents in Journal Club who actually was involved in writing the paper, it's maybe interesting to hear how, or the thoughts that we had when deciding on what experiment to do next, and how we had the ideas that we had. So this research was initiated due to a clinical findings in a patient, so called P1 in the paper, patient 1. She was a 4.5-year-old Swiss girl when she initially presented to our clinics, and actually she initially presented to us due to poor physical growth and sort of thin, sparse hair with discrete facial dysmorphia. And that was the initial reason why she came to the Children's Hospital, and she was being followed due to this sort of poor physical growth. And then at the age of 5, she came to our emergency room with very, very severe anemia. And what you can see, she was anemic, and what she also had is she had classical signs of an autoimmune hemolytic anemia. So her reticulocyte counts were very, very high, and her LDH levels were very, very high, as well as a sign of a bone marrow trying to produce enough red blood cells to compensate for the anemia. And the LDH is a sign of a lot of cell death. And what was interesting is that very, very quickly things changed. So her clinical presentation changed in the sense that she still had her persistent severe anemia, but her reticulocyte counts dropped very suddenly, as did her LDH values. So basically her autoimmune hemolytic anemia became more of a pure red cell aplasia. And she was also Coombs positive, so it really seemed to be an autoimmune hemolytic anemia. And she continued to be anemic over the following 5 years, during which time the study was conducted. And during these 5 years, she received various immunomodulatory treatments that all couldn't alleviate her anemia. And during this period, she required red blood cell transfusions on a regular basis. So she required red blood cell transfusions every 2 to 3 weeks for this 5-year period. She also developed a hypogammaglobulinemia with time and required immunoglobulins, which you see here. Now, of course, with this patient, what we very quickly wondered is what's going on in our patient's bone marrow, or what's the reason why she has this pure red cell aplasia. And a lot of this research will focus on erythropoiesis and hematopoiesis. So because most of you are most likely immunologists, I thought I'd just quickly talk a little bit about what goes on in the bone marrow. As many of you most likely know, stem cells that reside within the bone marrow give rise to cells of the lymphoid lineage, which we all know as immunologists very well. But in this case, we were more interested in cells that arise from the common myeloid progenitor. In our case, we were mostly interested in erythrocytes and platelets. But as you see, erythrocytes share a progenitor with the common myeloid share a progenitor with cells of the granulocyte and macrophage lineage. And when we look at a bone marrow sample from a patient, one of the ways that we can sort of evaluate how much erythropoiesis, or production of red blood cells, is going on is we can sort of see where the balance is between these two progenitor populations. So can a little bit see what the ratio is of the myeloid, or production of myeloid cells, towards the production of erythrocytes and erythrocyte precursors. And this is what we did in our patients' bone marrows. One of the first things we did is and we repeatedly did it throughout the years, is we looked at her bone marrow, and what we repeatedly always saw is we saw that she always had relatively normal, if not even slightly elevated levels of myelopoiesis within her bone marrow, but the erythropoiesis was always extremely reduced. And this is depicted here additionally as an increased myeloerythropoiesis ratio that would normally be around 3:1, and in her case was extremely elevated as a sign of reduced erythropoiesis. So as I mentioned, we wanted to see what was happening in the bone marrow. One of the theories that quickly arose is that potentially she had autoimmune infiltrates within her bone marrow that were attacking the red blood cell precursors. And if you look at sort of the immune histochemistry and histology on her bone marrow at the age of 5 years, what you can see is you can see lymphoid infiltrates that are CD4 and CD3 positive. And what's interesting is when you look at the flow cytometry, we can confirm that she has these CD45, CD3 positive infiltrates and has an increase in double negative T cells within her bone marrow. She also had a slight increase in double negative T cells in her peripheral blood, but the bone marrow was definitely more impressive. So just based on the looks of things, one could argue, well, this looks like this potentially could be autoimmune infiltrates, but we wondered whether maybe these are also just maybe lymphoproliferative infiltrates. And our question basically was, are these T cells actually attacking the red blood cells or the erythroid precursors in the bone marrow? And one idea we had to sort of answer this question as well as we could was to look at the clonality of the T cells within our patient's bone marrow. And I just really quickly want to just basically show you that what we found when we looked at the CD8 T cells in our patient's bone marrow is we found a very polyclonal infiltrate, similar in clonality to her peripheral CD8 T cells. And when we just looked at the IgH clonality, we also found a very polyclonal population, which indicated to us that what was going on in her bone marrow was most likely not only autoimmune. Of course, we couldn't fully exclude it, but it didn't look classically autoimmune. So we wondered, well, is there maybe another reason for her poor erythropoiesis? In parallel, we were also conducting genetic sequencing on our patient. So we conducted a whole exome sequencing on her and her family and found this de novo variant, or at this point it was a de novo variant, or a novel variant in STAT3 caused by a de novo mutation. And at this point, when we actually conducted this whole exome sequencing, there wasn't much published on STAT3 gain of function patients yet. So we initially thought this looks like a STAT3 loss of function mutation, very strange, doesn't really fit with our patient's phenotype. And we actually had to conduct RNA sequencing where we then found elevated STAT3 target genes. And then we finally noticed that actually what she has is a STAT3 gain of function mutation. Retrospectively, it's relatively obvious. I mean, she had elevated double negative T cells. She developed hypogammaglobulinemia with time. She developed hepatosplenomegaly with time. But at that point, it took us a while. But we were lucky to have the RNA data, or RNA sequencing data on our patients' PBMCs, because it also gave us some additional interesting information. So what you can see here is a scatter plot, and sort of everything above the dotted lines is significantly upregulated in comparison to a healthy control, and everything below the dotted line is significantly downregulated. And what was very interesting is that in our patients' PBMCs, we found that these hemoglobin alpha and hemoglobin beta genes were significantly downregulated. And what was also interesting is when we looked at sort of the hemoglobin genes in general, we also found that our patient had an interesting upregulation in hemoglobin gamma genes. And you can also calculate a ratio out of hemoglobin gamma to hemoglobin beta. And sort of in the sort of hematology field, this is sometimes used as a sort of proxy for how much erythropoietic stress a patient is under, and as a sign that our patient was under a lot of erythropoietic stress. And I'll come back to this ratio afterwards. All right, so we had our patient's mutation in STAT3. So of course, our next question was to try to find out what this mutant protein was doing within the cell. So just as a quick reminder for everyone, JAK signaling is a very straightforward intracellular signaling pathway. Cytokines or hormones bind to a cytokine receptor, phosphorylate JAKs, JAKs phosphorylate STATs, STATs dimerize, translocate to the nucleus, and then act as transcription factors in the nucleus while binding to various other transcription factors. So there's various parts along this very, very straightforward signaling pathway where you could imagine that a mutation could affect STAT signaling. So one of the first things we did is we transfected HEC293 cells with either the wild type or the mutant STAT3 and used a luciferase assay in order to quantify the transcriptional activity of either the mutant or the wild type STAT. And what you can very quickly see, this is on a logarithmic scale, is that already in resting conditions, the mutant STAT3 had an impressively increased transcriptional activity. What we thought was very interesting, though, is when we stimulated the cells with IL-6, we could push this increased transcriptional activity even further. And when using these two different STAT phosphorylation inhibitors, we could decrease the transcriptional activity as well. So this is relative to the resting wild type as a control. And this was interesting to us because it indicated to us that the mutant STAT3 wasn't constantly on at the same level, but that you could really, the activity was malleable and could be changed. So then, of course, we wondered what's the phosphorylation status, or does this mutation somehow alter the phosphorylation of STAT3? And what we found here is we very quickly found that the mutant STAT3 was causing an earlier phosphorylation of the STAT3 upon IL-6 stimulation, and was reaching higher levels of overall phospho-STAT3 levels. The next question we had, and this was a little bit through the collaboration that we had at this point with Stefan Ehl's lab, they were thinking a lot about, do STAT3 gain-of-function mutations alter how a phosphorylated STAT locates within the nucleus? So do you maybe with some STAT3 gain-of-function mutations have phosphorylated STATs basically just hanging out in the nucleus because they can't translocate correctly within the cell? So we wanted to look at this as well, and what we found in our patient was that there was no difference in the localization of the phosphorylated STAT3 between the cytoplasm and the nucleus, so this mutation didn't seem to be affecting the translocation of STATs to the nucleus. Then the next experiment we did was quite interesting, and I just like to take some time to explain this. So as I mentioned before, we know that STATs, they interact with with proteins within the cytoplasm and the nucleus. They're known to interact with each other, so they form homodimers, but some STATs can also form heterodimers, and some STATs can even form tetramers. So one of the questions we had is we wondered whether this mutation in STAT3 was somehow altering the way that the mutant STAT interacts with proteins. And the way we decided to look at this is we did pull-down assays with either the wild-type STAT3 or the mutant STAT3, and used mass spectrometry to evaluate which proteins were potentially binding to either the wild-type or the mutant STAT3. And let's just focus on the wild-type side of things first. What you can see is in the resting conditions, in the resting conditions, if you pulled down the STAT3, basically the only known or described STAT3 binding partner we found in resting conditions in the wild type was STAT3 itself. And then when we stimulated the cells with IL-6, we really only found STAT4 as a described known potential binding partner for STAT3. And this was very different in the mutant cells. So already in the resting conditions, we found S100A9, that at this point was described to be a potential binding partner, interaction partner, I should say, with STAT3, and this already bound in the resting conditions. And then upon IL-6 stimulation, we found 6 other described interaction partners with STAT3. And mass spectrometry data is quite often very messy. There's lots of potential binding partners that you find. And this data was really, really stringently filtered together with experts at the ETH of Zurich in this field. And the reason I'm saying this is basically after the stringent filtering in the wild-type STAT3, we really didn't even find potential novel STAT3 binding partners. But when we looked at the mutant STAT3, there were, I mean, an extremely impressive amount of potential novel or undescribed binding partners that we found in our pull downs as well. And this is just to say, I mean, you'd have to evaluate whether any of these are actually real binding partners, but it just indicated to us that there was much, much more interaction going on with this mutated STAT3. And if you look at where these various proteins sort of play a role, a lot of them play a role in inflammatory processes. So in summary, I mean, we don't need to go into detail with all these different proteins because they would all need to be evaluated. But basically, this is just to say that our mutant STAT3 was definitely showing very, very different interaction with inflammatory binding partners than the wild-type STAT3 was. So in summary, what we found in this initial part of the study was that our mutant phospho-STAT3 shows a malleable activity with an increased phosphorylation, although there's no preference between nuclear or cytoplasmatic localization, and we found indications of an increased binding of the mutant STAT3 to pro-inflammatory binding partners. All right, so, but what does this have to do with erythropoietin signaling and erythropoiesis? So I'm just going to quickly talk about what we know or what we consider to be happening during STAT3 or in STAT3 gain-of-function patients. So in STAT3 gain-of-function patients, we have an upregulation in STAT3 due to various mutations. And this causes an upregulation of various STAT3 target genes. Central among these are SOXs. So what we usually see is an upregulation in SOX3. And due to the overactivity of STAT3 or the mutation, SOX3 can't really downregulate the STAT3 activity. And due to the promiscuous activity of SOXs, we have an off-target effect on various other STATs, leading to a downregulation in STATs like STAT5 and STAT1, and an upregulation in STAT3. And what is known is that erythropoietin, which is the central hormone in erythropoiesis, signals via STAT5. So we very quickly thought, well, is it maybe possible that erythropoiesis doesn't really work in our STAT3 gain-of-function patient because of her downregulation in STAT5? So we had to get into looking at erythropoiesis. So one of the easiest ways to look at erythropoiesis is you basically, easily said, you take patient bone marrow, you take controlled bone marrow, you plate the bone marrow on a semi-solid medium, and then you count the either erythroid precursor colonies or the colonies, for here we counted granulocyte or macrophage precursor colonies. And what we very quickly saw is in vitro, our patient's bone marrow produced less erythroid precursors, as we expected. It's relatively difficult to work with cells on this semi-solid medium, so we wanted to develop an assay that would allow us to conduct flow cytometry, and that wasn't dependent on patient bone marrow, as this is more difficult to come by. So together with our colleagues at the hematology department, we developed a really nice assay that allowed us to culture patient PBMCs or controlled PBMCs, so peripheral cells for 14 days. And then via flow cytometry, we could analyze these cells. And what you see here is what these cultures look like after 14 days from a healthy control. So what you see is after culturing these cells, you have this sort of nice population of erythroid precursors that's formed. They're relatively large cells. And via their expression of CD71, which is the transferrin receptor, and CD235A, which is also an erythroid precursor specific marker, you can sort of confirm that these cells are erythroid precursors. The cultures also turn nicely red, so you kind of have a confirmation visually already that you have erythrocytes forming. And via the expression of these markers, you can also follow how cells are progressing through erythropoiesis. So they progress in this direction, then down here you'd have reticulocytes. Exactly. So this allowed us to, so with this, we had this nice assay that allowed us to really quantify the erythropoiesis in our patient. We could conduct this assay on bone marrow cells as well, but we didn't have very many samples. So we saw a reduction in erythroid precursors using bone marrow cells. That wasn't significant. With the PBMCs, we had more samples. So we could really nicely show that there was a really significant reduction in the erythropoiesis in our patient. And one of the initial findings that we had is we could show that the distribution of cells throughout the erythropoiesis or throughout erythropoiesis was similar or comparable between the patient and the healthy control, indicating that there was just an overall reduction in erythropoiesis in our patient and not a stop at some point during the maturation of the red blood cells. Then the next thing that we wanted to do is we really wanted to just confirm that what was causing the reduction in erythropoiesis in our patient was actually the STAT3 mutation. So we virally transduced control bone marrow cells with GFP expressing virus expressing either the wild type or the mutant STAT3. And what we could very quickly find here is that the mutant STAT3 was indeed causing a reduction in the production of red blood cell precursors. All right, so now we had our assay set up. We knew that it was STAT3 causing the reduction in erythropoiesis. Now we wanted to see whether we could show that there was actually a defect in erythropoietin signaling. So now we had our assay that we could do flow cytometry on. So we used phosphoflow to look at the phosphorylation levels of phosphostat3 and phosphostat5 within our patient or the control cells. And what you can see here are phosphostat5 levels. In red is the mean fluorescent intensity of the mutant STAT3, and in black is the wild type STAT3, and you can see the various erythrocyte precursor subsets from the bottom to the top. And what you very quickly can see is that there wasn't really much of a difference in the overall phosphostat3 levels between patient and control. But when we normalized these to whole STAT3 levels, we found that there was a clear significant upregulation in the phosphostat3 levels in our patient. And what was more interesting was that what we also clearly found is that the phosphostat5 levels were clearly reduced in our patient's cells upon erythropoietin stimulation as an indication that the phosphostat5 signaling wasn't really working. Now, I'd just like to take a quick moment to talk about binary and graded signaling in erythropoietin signaling via STAT5. And the reason I just like to quickly talk about this is that I think it gives you an important idea of why we thought these findings in our patient were so interesting. So this comic nicely depicts what binary signaling or what binary anything is. So binary is basically on or off. Now, the group or from this paper that I mentioned here did a lot of very interesting research on STAT5 phosphorylation downstream of erythropoietin. And what they nicely could show is that when you have an erythropoietin signal, you initially actually have a binary on-off switch within a cell. So basically, you only need relatively low levels of phosphostat5 in order to turn the switch on, and you only need very, very low levels of phosphostat5 to uphold erythropoiesis under normal conditions. The time when your cells need high levels of phosphostat5 is when you require a graded cellular response in response to high levels of erythropoietin. So when you have states where high levels of erythropoietin are present, for example, during stress erythropoiesis, then you need high levels of phosphostat5. So if you have a model, and they had a mouse model that showed this, but one could argue that this is what we see in our STAT3 gain-of-function patients as well. So if you have a model where this sort of graded response is stunted, then you would have a basal erythropoiesis that would work well, but the stress erythropoietic response would be stunted. which of course has interesting implications for patients under erythropoietic stress, such as autoimmune hemolytic anemia, for example, as we see in our patients with STAT3 gain-of-function mutations. So I've told you about one patient. We managed to, via various collaborations, collect samples from 4 other STAT3 gain-of-function patients. And in all of these STAT3 gain-of-function patients, interestingly, we found a reduced erythropoiesis that was present. So this kind of confirmed to us that what we were seeing in our patient wasn't just unique to our patient, but was more universal. And what we also found in all of our patients, interestingly, is that they all had elevated erythropoietin levels as an additional sign that these patients were actually under, that's these STAT3 gain-of-function patients, were all under some form of erythropoietic stress that they were trying to respond to. And just this last little bit of data, what we also looked at in the patient PBMCs that we received is we wanted to see which cells in their PBMCs were producing red blood cells. And one thing we did is what we know from the literature is if you take control PBMCs and you sort out the CD34 population in PBMCs, so this should be the cells that are sort of stem cell-like, you actually see a higher production of erythrocytes from the CD34 negative population in PBMCs, which one doesn't really fully understand yet, but this has been repeated by various groups already, and this is just something that is easily reproduced, and we could reproduce it as well. And what was interesting was that in our STAT3 gain-of-function patients, it was the exact opposite. So if you actually took their CD34 cells, sort of the stem cell-like cells from their PBMCs, they showed a normal erythropoietic capacity. And it was only the CD34 negatives, or it was the CD34 negative population that was giving rise to this reduced erythropoiesis, which is interesting because then you, of course, wonder what causes this, or what triggers a reduced erythropoiesis in the cell. Is it some kind of stimulus that the patients had during in vivo, some kind of inflammatory stimulus? We don't really know, but there's active research going on this. All right, so this is the last bit of the story. So based on our findings, we had the theory that our patient was suffering from this severe anemia because of a faulty erythropoietin signaling in her cells. And what we decided to do at this point is treat her with a JAK inhibitor, so ruxolitinib. And what we saw after we started her on this JAK inhibitor is we, for the first time in the 5 years we've been following her, we saw a spontaneous increase in her hemoglobin counts, or in her hemoglobin levels, without a transfusion. But at the same time, what we also saw is a drop in we saw a thrombocytopenia in our patient. And because of this severe thrombocytopenia, we had to discontinue the JAK inhibitor therapy and place her on high-dose steroids. And then after her thrombocytes rose, we placed her on cyclosporin and tocilizumab, and she's now currently under this therapy. And as you can see indicated here, her hemoglobin counts stabilized, as did her thrombocyte counts during this period, and she's now absolutely transfusion-free. So for the first time in 5 years, we managed to normalize her hemoglobin counts, and she's not anemic anymore. And what was additionally very interesting is after she'd stabilized, we received PBMC samples from her once more. And at this point, when we conducted our erythropoiesis assay, we saw a normal erythropoiesis. And what was even more interesting is when we repeated the RNA-seq on her patient's PBMCs, we now saw a normalization in STAT3 target genes, and also a normalization in this hemoglobin ratio that I mentioned at the beginning of this presentation. So just an additional indication that her erythropoietic stress had been alleviated. All right, and that's the end of the story. So I'd just quickly like to say thank you to all the people who were involved in this work. Big thank you to everyone at the Pediatric Immunology Department at the Children's Hospital Zurich, and our colleagues at the Hematology Department who really helped us in conducting this work, our colleagues at the Functional Genomics Center and at the Immunology Department at the University Hospital of Zurich, then our friends at the ETH of Zurich who really helped with this work, and our friends in Lausanne who sort of helped from the hematology side additionally, and then to all of our various collaborators, so Stefan Ehl's group in Freiburg, and Anna and Adam in Prague, who helped us establish our collaboration in Greece and helped us attain all these patient samples. And then I'd just like to leave you with a little fun comic, and I hope this inspires a few of you to ask some questions, and thank you very, very much for your attention.
Thank you very much, Andrea. So, everybody, who has